It's a real pleasure to welcome you all to our second Stuart A. Northrop Distinguished Lecture. And Corey is going to introduce our speaker here in just a second. But I thought I'd take a few minutes just to recognize and acknowledge Bill Lovejoy here, who's the donor that started this Distinguished Lecture series and gave a nice, generous donation to the department. And we're going to keep this going for many, many years. Bill was here last year for our inaugural lecture, and we're really delighted to see him back here again. I saw him yesterday and said, "We're wonderful to see you here, Bill." And he said, "It's wonderful to be anywhere." And I thought, yeah, that's true. So I thought I would just say a few things. You all got the handout for today's lecture that describes our distinguished lecturer, Dr. Susannah Porter. And on the back is a little bit about Bill. But I thought I'd say just a few words about Bill, if I may, before Corey starts with her introduction. He's a professor emeritus of biology at Georgia Southern University. He comes from Ohio originally, went to, let me make sure I pronounce this correctly, Muskegon College. It's now Muskegon University. And he has endowed 4 separate lectureships around the country with institutions that he was affiliated with, including the John Glenn Distinguished Lecturer at his alma mater, the Robert Storm Distinguished Lecture Series at Oregon State University, the Darwin Lyle Distinguished Lecture Series at Georgia Southern, and the best one at all is the Stuart A. Northrop Lecture right here in EPS. And so we're very grateful that you were able to make this donation and make this possible, and we're also really grateful that you were able to be here and participate. So that's fantastic. Yeah. And I also wanted to point out one other thing. We have a couple of Caswell Silver board members here, including Pat Gratton, who's sitting halfway up. And Pat was a student in one of Bill's mineralogy classes. Bill was his TA, and Pat actually met his wife, Jean Marie, in that class. And they were actually together here in Northrop Hall 65 years ago, and they ran into each other for the first time last night. And so they had a lot of catching up to do. So that's pretty cool. With that, Corey, please take it away.
Awesome. Well, I will echo that thanks to Bill for making this lecture series possible. Like Peter mentioned, this is our second Stuart A. Northrop lecture, distinguished lecture. And I'm really happy to introduce our speaker this year, Dr. Susannah Porter. She comes to us currently from University of California, Santa Barbara. Really happy, sunny place, although we have pretty good weather right now, too. She did an undergrad, I'm only mentioning this because it was interesting, in mathematics, and saw the light and went to graduate school in paleontology and hasn't looked back since that time. She was a graduate student at Harvard University under Andy Knoll, who turned out to be my postdoc advisor. So we have, small world connections there. She went on to do a NASA astrobiology postdoctoral fellowship before starting at UCSB, and that was at UCLA under Bruce Runiger. And then ended up at UCSB and has been doing amazing things since then. So she is a professor there now. She's also vice chair of the department and the associate director of the Earth Science Institute at UCSB. Has advised several excellent students and won several excellent teaching awards at both the graduate and the undergraduate levels. So I'm really excited for a, very vibrant lecture here. And I should say, Susannah is also extremely famous in the paleontological world. She was the first person, or I should say, she discovered the oldest and first evidence of predation in the fossil record, and I really hope that we'll hear a little bit more about that. So, with not too much ado, Susannah.
Thank you. Thanks so much, Corey. Yes, you will hear about some of my work on early predation. I realized when I was putting my slides together that I missed a really great opportunity to have a sort of a horror movie double feature and call this Tiny Vampires and the Living Dead rather than Living Fossils, the record of early life in the Grand Canyon. But I'm just to sort of lower your expectations a little bit, I'm not actually going to have any fossilized versions of Nosferatu here, or zombies. The organisms I'm talking about look more like this. Much tinier, in fact, invisible to the naked eye, but I can assure you if you were a single-celled organism living at the same time as these, they would be absolutely terrifying to you. I'll show you why later on in the talk. So my talk today is going to be about fossils that I have studied over the last 20 years or so in the Grand Canyon, from the oldest fossiliferous rocks that we know of so far in the Grand Canyon. And these provide a glimpse of what life was like 200 million years before animals really take off in the Cambrian explosion. There are no animals that we know of at this time, but the ecosystems, as we'll see, were pretty complex. And that complexity, I'm going to argue, maybe can give us some insight into why we subsequently had the rise of complex life like animals and seaweeds 100, 200 million years later. But before I get to that, I wanted to just give you a brief overview, because I know not all of you maybe know everything about the early fossil record of life. And I actually want to start by pointing out that this is a really young science. So if I were giving this talk 70 years ago, I would basically say, well, the oldest definitive evidence of life is Cambrian in age. And there are these structures known as stromatolites that a lot of people think probably are signs of life, are algal mats, but it's indirect evidence. There was, at that time, no body fossils that had been found that were convincingly Precambrian in age. Actually, since then, looking back at some of the early reports, we recognize, oh, maybe people were describing some Precambrian body fossils, but they didn't realize it at the time. So that all changed. And I should say, by the way, this is not too different from what it was like during Darwin's time, and he famously fretted over this lack of a Precambrian fossil record on the origin of species, and what his theory predicted that there should be ancestors to the trilobites deeper down in the strata, and was concerned that we hadn't found any. So that all changed in the 1950s and 60s when this report in particular, Tyler and Barghorn, 1954, in Science. Tyler was an economic geologist, and he was studying the Gunflint Formation up on the shores of Lake Superior in Ontario, and he collected some rocks that he thought maybe were coal. He sectioned them, and he found that they were filled with these complex structures. And so he sent them to a paleobotanist, also Barghorn, at Harvard, who's actually, turns out, my academic grandfather. And Barghorn and Tyler published that first paper, and then later a more detailed paper in Science showing this incredible diversity of organisms. I mean, and it was unquestionable. No one doubted the age at that time. 2 billion years, a little bit younger. We know now, 1.9, 1.8, and the fossils were clearly part of the rock, and this great diversity of microbes, of bacteria. Around the same time in the Soviet Union, Boris Timofeyev was taking shales and siltstones and sticking them in hydrofluoric acid and looking at the organic residue that resulted from it and reporting organic walled microfossils of uncertain affinities that are flattened, compacted in the shales, and named a whole bunch of different species, many of which are still valid today. So the importance of these two lines of research, the Barghorn and Timofeyev, was not only that they showed that there was indeed there were indeed life 2 billion years ago. They also showed paleontologists how to look for it. So we had a search image. We knew to target black cherts, where we could find three-dimensionally preserved organic walled microfossils, and shales that we could dissolve in acid and get out these flattened organic walled microfossils. And so since that time, so paleontologists went out and sort of sampled all over the world, and reports were coming in of these body fossils. Since then, the fossil record of the Precambrian has expanded a lot by looking at chemical fossils. And two types in particular, stable isotope ratios, stable isotopes of carbon and sulfur and iron, that can tell us about ancient metabolisms, the presence of ancient metabolisms, And biomarkers. So these are ancient lipids. So they're derived from like cell membranes of ancient organisms that have been altered through temperature and pressure to more stable compounds, but we can infer often which specific taxa that they are derived from. So if we put this all together then into what we now know, 2018, about the history of life, what I think is amazing is that we have definitely evidence of life 3.5 billion years ago, and it's actually fairly diverse in several different environments. Possible evidence of life greater than 3.7 billion years, and there are, by definition, no rocks of Hadean age. And so I think it's amazing that basically the first time we could possibly preserve fossils of life, we see it, right? So what does that mean about how likely the origin of life is in the universe? I'll let you speculate. And then, following this, bacteria and archaea, so simple cells, prokaryotes, they diversify quite rapidly. And we know by 2.4 billion years ago that life had expanded across the planet, lived in all sorts of different environments, lakes, near shore, shallow waters, deep hydrothermal vents, and had a variety of different metabolisms, effectively eating hydrogen or hydrogen sulfide, breathing iron oxide, and so on. And cyanobacteria, the source of all of our oxygen, almost all of our oxygen on the planet, had evolved as well. Then starting 1.6 billion years ago, we start to see evidence of more complex cells. Our ancestors, the eukaryotes, they appear and they diversify. And what's interesting, getting back to Darwin's conundrum, is that the oldest evidence of animals is pushed back older to during Darwin's time, but it hasn't been pushed back that much. There's evidence from biomarker compounds of maybe sponges 650 million years ago, but really nothing convincing older than that. From the fossil record, molecular clocks maybe would give you a different story. So this is a very simplified history of life. Life appeared very early, but it was microbial for most of its existence. For 83% of the history of life on Earth, it was a microbial planet. And complex organisms only appeared, in the last 17% of life history, 13% of Earth history. And this actually makes sense the more we learn about the diversity of life on Earth. So I don't know how many of you remember your high school biology class and what you learned, but this is what I learned in my high school biology class. I talked to my mother. She learned that there's two types of life, animals and plants, and everything fit in that. And I learned there's 5 kingdoms. There's the plants, the fungi, the animals, the protists, and the monarans. Right? But actually, my biology class was pretty out of date because in the 1970s, and I went to high school in the 1980s, in the 1970s, it had already been discovered that actually, rather than 5 kingdoms, there were 3 great domains of life. The archaea and the bacteria, which are microbes. They look basically like little balls and rods and squiggles. And then the eukaryotes, where the protists, that includes the protists. And then plants, animals, and fungi are tiny twigs at the tip of this tree. And moving fast forward to today, that picture becomes even more humbling, I guess, in that we realize the red dots represent just organisms we know from sequence data, so they haven't been isolated. We've never seen them. And what they show, I know you can't see the words here, but basically these are the bacteria. This is the candidate phyla radiation, it's called, closely related to the bacteria. These are archaea. So this is a group that if you looked at it, it's a kind of prokaryote, doesn't have a nucleus, looks very similar to bacteria, metabolically pretty different. And then this green thing are the eukaryotes, protists. And animals, they're not even a twig in here. I mean, they're just a little branch. Right? In terms of genetic diversity, There's so much more out there than we appreciated, 50 years ago, 100 years ago. Okay, so my research, I got into paleontology because I was fascinated with the Cambrian explosion, and I was interested in the origins. How did complex organisms arise? How do we go from a microbial world here to a world teeming with all the beautiful complex organisms that are around us? And I thought to myself, well, part of my research is on the Cambrian explosion, some of these weird early animals. But also, if we want to understand the origins of the Cambrian explosion, it makes sense to look back in time to the common ancestors of the animals, to the larger clade, the larger group that animals are a part of. Because actually the Cambrian explosion is, we focus on animals, but there's other stuff going on at that time with seaweeds. There's diverse seaweeds appearing at this time as well. So there's sort of a bigger phenomenon going on among the eukaryotes. Okay, so that's what my research focuses on. And again, I realize I've just to make sure we're all on the same page when I talk about prokaryotes and eukaryotes. Prokaryotes, again, those are the organisms that don't have a nucleus. They don't have organelles like mitochondria or plastids. Eukaryotes, on the other hand, are more complex cells with nuclei. Some of them have plastids where photosynthesis occurs, like in plants and algae, mitochondria. And again, just to emphasize, when we talk about eukaryotes, they include the plants, animals, and fungi, but the vast diversity of eukaryotes are protists, are single-celled organisms. And maybe you're like I was when I started out my PhD, and I thought, oh, protists, they're kind of boring. They're just like simple little animals. And now I realize, 20 years later, how wrong I was, how fascinating this group is, and how, if you wanted to be like a Victorian-era naturalist exploring the Amazon and finding new species of birds, well, that's probably mostly done by now, but there is so much to find among the protistan world. That's where you should be focused if you want a life of discovery like that. Just to show you a couple of my favorite, before I get into the Chuar Group, this is a paper that describes this amazing sort of cooperative, helpful behavior by amoebae. So in this culture, there are amoebae that are, undergoing mitosis, right? Creating a daughter cell, and about a third of them get stuck. They don't do that final split. And these authors showed that the amoebae, when they're struggling, they release this kind of chemical help signal, and a nearby amoeba, in this case the green one, will come along and insert itself and help split them apart. So they refer to them as midwife amoebae. So this fascinating cooperative behavior. Here's another wonderful example of sort of the complexity of this group. These are our close relatives, our closest protistan relatives, the choanoflagellates. A number of them make these very thin glass rods that they weave together into these beautiful baskets. And so there's a picture of a basket, and then here on your right is where they took one of these poor guys and dissolved it in hydrofluoric acid. So the glass is gone, but you can see these are extensions, the pseudopods, the fingers of this cell that are used to weave together this basket. So early basket weaving. And then this is the most stunning, I think. This is reported in the 60s. A dinoflagellate, so these are the same group that includes the zooxanthellae that live in corals, right? And also the ones that are responsible for red tide, the poison, the toxic shellfish that make this toxin. They're also responsible for the bioluminescence that if you've ever sort of splashed around in the ocean and lights up, it's these organisms. So this is a group of dinoflagellates that apparently has a subcellular analog of our eye. So it has the equivalent of a lens and a cornea and a retina, which apparently is some endosymbiont, another sort of photosynthetic bacterium in there. But it functions like an image-forming eye, like ours. And so the question is, well, where's the brain? Like, why does this organism need to see an image? Apparently it does. So there's still a great mystery there. Okay, so let's move to the fossil record and my work. So, the oldest eukaryotes, this is always sort of bouncing around. If I gave this talk 10 years ago, I'd say there's evidence 2.7 billion years ago. I'll say the definitive evidence that I'd say is widely agreed on is that by 1600 million years ago, 1.6, we had eukaryotes of some kind. And these are the fossils that tell us that they were around. They're microscopic, but they have characteristics that are not known among the prokaryotes. They have irregular branching and budding, this sort of complexity of this organic envelope. Here you can see this is these concentric circles, like lines of latitude that go around the cell body. So it's pretty widely accepted that this level of complexity suggests that they are eukaryotes. What kind of eukaryotes, though, we don't know. I should say, feel free to interrupt and ask questions. Around the same time, a little bit younger, this is a fairly recent report of macroscopic, decimeter scale organisms. And again, so we can't necessarily tie it to a particular group of eukaryotes, but we just can say, well, we don't know of anything like this evolving in prokaryotic organisms, and so it makes the most sense to suggest that they're probably eukaryotes of some kind of like modern seaweeds.
Are they single cell or
multi-cell? Multi-cell is the argument. And it's hard to tell from these, but these authors did a maceration, and they got some organic material out that's multicellular. Rocks about the same age. Again, a macroscopic organism. It's some beautiful specimens here from India, where I don't know if you can make it out, but they seem to be septate, which suggests sort of, again, a level of complexity and the size together suggests, well, this makes sense with what we know about eukaryotes rather than prokaryotes. But we don't really know what kind of eukaryote it would be. So if we move to now rocks a little more than 1 billion years old, we have the first fossils that we can confidently assign to a living group of eukaryotes. And that's this very special fossil, Bangiomorpha pubescens. Has only been reported from one place, Arctic Canada. And here are the fossils. You can see there are these filaments, and the filaments are characterized by this unusual cell division, where we have cells that divide, and then each of them divide, and they sort of have this pill-like shape. And then the clincher is that this type of further cell division that is along the longitudinal axis we see here in the fossils is just the spitting image of what we find in this modern red alga, Bangia, which you may have seen it if you've walked along a rocky seashore, say in the Pacific Northwest, a little red fuzz on rocks. And these, in fact, have attachment holdfasts where we can see, I don't think I have a picture here, but the original surface that they were attached to as well. So here we can say, this is exciting because we can say, okay, we know that there was branching within the modern eukaryotes by this time. So what that means is characteristics that are associated with all eukaryotes today, the nucleus, the mitochondria, sex, cytoskeleton, we know that had to have evolved by then, right? Because we have clearly a group of red algae by this time. Okay. And then if we sort of move now from 1 billion to 800, 750 million years ago, we see evidence for diversification, for expansion in the eukaryotic clade. So this is taken as the time when eukaryotes really sort of rise to prominence, protists rise to prominence. And the lines of evidence include the diversity of these organic walled microfossils, primarily in shales. If you remember what Timofeyev was doing, dissolving the shales in acid and getting these flattened organic walled fossils out. So these bars here indicate the sort of uncertainty in the age of these different fossil assemblages, and then but the diversity within each one. And you can kind of see that, yeah, there's an increasing diversity here, a gap here. You can guess what that is. I'll tell you in a minute. We also see around this time an evidence for more and more multicellular forms. We don't necessarily know, again, where they fit in the tree, but they're clearly sort of evidence of increasing complexity. Also, at 810 million years, we have evidence for, it's the oldest evidence for controlled biomineralization in life at all, or among the eukaryotes. Or maybe there's some bacteria fossils that make magnetite or something. But these are these gorgeous apatite scales. There's 36 or more species that have been described from just one locality or one place, Yukon, Canada. And it's thought that these scales were perhaps like modern coccoliths, that they armored a protestin cell. And you can see in some cases, they not only covered it, but they had these spines sticking out. You can see that there as well. We'll come back to these again later. So again, back to this evidence for expansion. Morphological innovations, biomineralization at 800 million years. And then something I'm going to talk more about, evidence for, again, kind of similar to biomineralization, but organic tests, that these houses that the protists built around their cells. Snowball Earth accounts for that big gap in the diversity record. So starting at 720 million years, there were 2 snowball Earth glaciations up to 635 million years where there's almost nothing known about the record of life. And then, of course, I'm not going to get into this today, but on the heels of the snowball Earth glaciations, then we have the Ediacaran period and the expansion, the appearance of macroscopic, diverse life, and the Cambrian explosion of more recognizable animals. Okay, so we're going to move to the Grand Canyon now and focus in on the Chuar Group. And the Chuar Group, notably, actually should be higher now. But it's sort of this last glimpse of eukaryotes just when they're sort of at their most expansive, right before these glaciations. Anyone have a question while I take a drink of water? Okay, so moving to the Grand Canyon now. I had the great privilege as a PhD student to go on 2 river trips led by Carl down the Colorado, and we hiked up, and hopefully some of you have had this opportunity to go to the Grand Canyon Supergroup. So just to situate you here, we're in the Chuar Group, which is the upper part of the Grand Canyon Supergroup. And this is a picture of what it looks like, courtesy of Carol Daler. Some of the fossils I'm going to talk about come from these gorgeous sediments. The Chuar is actually a really valuable unit from the perspective of sort of understanding Neoproterozoic, mid-Neoproterozoic Earth history. It's one of the best dated units. You can see this is from a recent paper. Many of these dates that give us a really great age constraints. And I'm maybe not that objective, but some of the most beautiful fossils from this time as well. And this is maybe what it looked like. So it was a shallow, tropical, quiet seaway. If you were alive back then, you probably wouldn't have been able to breathe. But the things you would, if you go scuba diving, the evidence of life that you would be able to see are the stromatolites. There are stromatolites in the Grand Canyon. There are actually the largest critters around are 1 millimeter, 2 millimeter size, Chuaria circularis. Everything else is microscopic. So my talk today is going to focus on these microscopic fossils. I'm going to first talk about these vase-shaped microfossils, or VSMs, and that's what inspired the Night of the Living Dead. Really, I called it living fossils because they look so similar to some modern groups. And then I'll move on to talk about these organic-walled microfossils that are abundant and diverse in the lower part of the Chuar Group. And I'm not saying that they themselves are vampires, but we see evidence, as I'll show you, of vampire-like predation on them. Okay, so let's focus on the VSMs first. And I just want to point out actually how important this fossil group is. This is a recent estimate of eukaryote diversity. that is done using this constrained optimization method. If you don't know what I'm talking about, don't worry. But basically, it's a way of calculating diversity through time of rather than binning fossil assemblages. And the reason I show it is just to point out, here, this diversity is organic walled microfossils. But it's declining here. Here we have the glaciations, right? There's very little known about this time right before the glaciations. But right here is this VSM peak. Okay, so these vase-shaped microfossils are known from all over the world. They're often quite abundant. They're, I think, known from 14 different assemblages all over the world, which is a lot for the Precambrian. And they're the only guys around. So, it's almost as if it's a vase-shaped microfossil world at this time. Those are the only fossils that we have. They're all over the place. They're abundant. They're the last evidence of life, and then we go into the glaciations. So I think it's telling us something, but I don't know what. Okay, so the vase-shaped microfossils, they were first reported from the Grand Canyon by Bonnie Bloser, and they got the nickname Bonnie Bags. She was a graduate student at UCLA who worked on them, did some really great work. Her fossils were from the shales, the upper part of the Chuar. And then when I was a graduate student, my advisor, Andy Knoll, went on an earlier trip and went to 60 Mile Canyon and collected, I guess this nodule is referred to as the Andy Knoll nodule, apparently. So you can get a sense of how giant it is. So he collected this nodule and a bunch of shales, and he brought the rocks back to me, and I was a second-year grad student trying to figure out what to do with my life. And he's like, here's some rocks. Why don't you just see what's in there? And actually, I think for this nodule, he was like, send him off to the geochemist colleague. And I got mixed up, and I didn't send him to the geochemist. I did paleontological analyses on him instead. But I'm glad I did, because what I found when I thin-sectioned it was just absolutely full of these vase-shaped microfossils. Just next, cheek by jowl stuffed in there. And so calculate, there's lots of, look at the size of this thing. There's lots of nodules all over the top of the Chuar. There's got to be trillions of these fossils available. That's 0.5 millimeter there. We can study them in thin section and learn a lot about their various shapes. But we were also very lucky to find that we could dissolve these nodules, which are made of carbonate, in acetic acid and get out these beautiful glass vases. And they look like little see-through vases that you could stick little tiny flowers in. So I did a bunch of SEMing, looking at these guys. And I'm just going to quickly show you some of the different species. And you don't have to worry about the names, but just some of the different characters that you see. So this species is characterized by this perfectly equilateral triangle aperture, this opening in the shell. This species is characterized by having 6 indentations around this aperture. You can see here that hexagonal aperture when you're looking straight down on it. Circular on the inside. This is a cross-section through here, so you can see it's an invaginated aperture. These are 2 pretty simple species that are characterized by a circular opening or aperture. One species has a collar around the rim. Here's another species that has a sort of lobate or flower-like aperture. You can sort of make it out there. Bonnia, named for Bonnie Bloser. It's characterized by having a curved neck and sort of different, shorter curved neck versus longer curved neck. And I should point out, this is my favorite species name that I came up with. I had a friend in grad school who studied ancient Greek poetry, so it was perfect. She helped me out with the Greek and Latin names, and I was telling her what I was studying, and she said, there's this Greek word, dakrukares, which means to derive joy from tears. And I thought, oh, that's perfect. That's perfect, these tear-shaped fossils. Bombycian, these I only have in thin section, but here you can see the aperture is inclined, sort of flush with the wall, no neck. This one, which is, Again, I just have a thin section, but I can tell from focusing up and down that it's hemispherical in shape, and it has a circular aperture, slightly invaginated. Circular aperture there. This one, again, quite rare. I only know from thin section, but I suspect this is a cross-section through it, and then these are longitudinal sections. You can see it's indented, and that the aperture, the opening, is probably somewhere in there. And then this, finally, this strange species that I'm still trying to make sense of. I named it after honeycomb, Melly, honey, where this black that you're seeing is a pyrite coating, and then it has these regularly arranged, regularly sized holes in the wall. Oh, I forgot to mention one puzzling specimen, maybe agglutinated, I don't know. So this is back in the days before there was any sort of digital camera, so I took a bunch of pictures, made them in the dark room, sent them off to all the different protistan colleagues I knew of by snail mail, and I got answers from some of them, and they said, that looks a lot like a testate amoebae test. And sure enough, the more I dug into the literature of testate amoebae, I could see so many examples where they looked so similar. So what are testate amoebae? It's just like what it sounds. You guys have probably heard of amoebae, right? And you've probably seen pictures of naked amoebae, just the sort of wiggly shaped things with their pseudopods extending. Well, there are also amoebae that actually build their own shells, like a snail builds its own house. But actually, remember the choanoflagellate with its fingertips weaving the baskets? It's the same kind of thing here, where the cells will use their fingertips to sort of shape their test. There's two different types, the euglyphid testate amoebae, and they have this more phylos-like pseudopods, filamentous, and the Archalinida, and they have these more lobos type filaments. And strangely enough, they're in very different parts of the eukaryotic tree. So here we are, by the way, just to situate you, we're in the piscicons, animals, fungi also in there. Here are plants and algae. Those archelinids, the lobos form, are over here, and the euglyphids are over in the other part of the tree. This is an unrooted tree, so we don't exactly know how the relationships of these different groups are, but suffice it to say that these tests are not homologous. They're not present in a common ancestor. It's an independent evolution of tests. But these tests are gorgeous, and I could have spent hours on this slide coming up with different examples, but I just showed some of my favorites. So there are some that agglutinate, so they pick up particles from the environment, and they build their tests with that. And in some cases, they'll even use, you can see some diatom skeletons in there. Sometimes amoebae will eat other testate amoebae and take the particles of their test and build their own test. In some cases, the amoebae will mineralize their own structures and fit them together. You can see this here. Here's an example of square plates that this amoeba has fit together. These are more circular plates or ellipsoidal. This is an example of ones where they've eaten somebody else and taken their plates and stuck them together. And then there are forms that are just organic, entirely organic. And I should say these guys, those are fit on top of an organic test underneath. All right, so just to convince you, I think these fossils, the VSMs, are testate amoebae. I'm going to show side-by-side modern analogs here, and then fossils, so the species you've already seen. So you remember this triangular aperture form? There's nobody in the modern that we know of, in the modern world, that looks exactly like that, but there's certainly this ability to make triangular apertures is present in the archelinids, and actually restricted to this genus. That hexagonal sort of appearance aperture and the invaginated aperture, This is known today in the archelinids, again. The teardrop shape, this is actually a pretty common form. It's found in the euglyphids, archelinids. We also see it in single-chambered foraminifera. That lobed aperture is also found in both euglyphids and archelinids. The curved test, Bonia, that's also found in both euglyphids and archelinids, so convergent evolution. That inclined aperture, also found in both euglyphids and archelinids. That hemispherical shape, that is uniquely found today in this one genus, Archela. And this shape, which I think is polygonal in cross-section and these indentations, that also is known today just in the archelids. This honeycomb type, which I said, I'm not really sure how to interpret it. At the time we published this paper, we thought, well, maybe what's going on is this is where the bases of these scales are embedded in an organic wall, similar to what we see today in euglyphids. I'm not so sure about that anymore, but if that interpretation is correct, that character is only known in the euglyphids. I also didn't mention, so these VSMs that have been found elsewhere in the world, there's a great deposit in Sweden that has a number of vase-shaped microfossils where they have these circular bodies inside. And lo and behold, in testate amoebae, when times get tough or it's time for have sexual reproduction, they will make a cyst inside the cell. And so I think that's what's going on here. In the Uinta mountain group of Utah, same age as the Chuar, I found a specimen where it looked like they were sort of mouth to mouth. This looks very similar to what we see today in modern testate amoebae, where the mother cell will first form the test of its daughter cell and then undergo cell division. And they're mouth to mouth, just like that. Okay, so one of the questions I struggled with is, okay, this seems like a great fit. They're so similar. But the fact that we're seeing these tests today in two very different groups in different parts of the tree makes me worry a lot about convergent evolution, right? So could it be that back during Chuar time, this is yet a third group of protists that evolved these tests independently and they went extinct? And I suppose that's possible. You sort of have to use Occam's razor, right? And say, well, okay, what's the simplest explanation? Is perhaps that we have evidence of one of these groups, at least back at this time. So this curved test, if you remember, I mean, it's present in both euglyphids and archelinids, but there are some species of VSMs where they show characters that are only known today in the archelinida. And so I think I have a fair amount of confidence that probably this is the group that was around back then, 730 million years ago. And actually, molecular clock analyses that try to date when different groups diverged suggest that this group could very well have been around. Euglyphids, as I said, I'm not, at the time we published the paper, we thought, oh, we have evidence for this other group as well. I'm going to step back from that a little bit and say I'm not so sure. Other VSM species, maybe, who knows? Maybe they're archelinids is probably the safest thing to suggest. So I think we can say, okay, we had the red algae at 1 billion years ago, and now by 730 million years ago, we see evidence for branching within some of these major clades of eukaryotes. What's interesting about the Chuar group, though, is, and a lot of these other records of VSMs, is that we think that there's evidence that it's marine. And all the species that I was comparing these to are from terrestrial or freshwater environments. A lot of them are from sphagnum moss. So we have this interesting thing where, okay, we've extended the fossil record by basically doubling it, but it's in a very different environment from where they are today. So it suggests that, well, maybe we had an early diversification in the marine, and they moved on to land. And there are other examples of that from the fossil record, where modern velvet worms, we know, have this Gondwanan distribution known in Australia, for example, only terrestrial. And in the past, they were quite diverse in the Cambrian in the marine realm. So, one possibility is that they're maybe still around today in the marine realm. It's kind of undersampled. And of course, the terrestrial freshwater record of the Precambrian is not very well sampled. But maybe they did make the conversion onto land following land plant evolution and disappeared in the marine environment with the radiation of benthic forams, taking over their niche. And, it's interesting to note that there's no record, snowball Earth glaciations show up right after they appear, and they're gone after that. So maybe that did them in, and they just survived in refugia. So why is this important with this talk? Well, if these really are testate amoebae, we know that modern testate amoebae are voracious predators. They eat not only bacteria, but other protists, each other, and they even eat animals. So this is an example of a testate amoeba that's coming up on a rotifer, reaching out with its pseudopods, grabbing it and sucking it in. So we think, okay, these guys were probably predatory as well. So let's move on and talk about evidence for vampires then. So I mentioned earlier to some of you, when I started working on these organic walled microfossils, or when I started working on the Ma Chuar Group, I said to my advisor, well, the organic walled microfossils, there's already a paper on them from the 1980s. That's already been done. There's nothing more for me to do. And I realized how naive that statement was now. So I took another look at these fossils, first with the light microscope. And I was just really frustrated with using the light microscope because, I don't know if you can see this here, but there's sub-micron details. I could tell that they were there, and there was something interesting. But with the resolution of a light microscope, I just couldn't see what was going on. And these fossils are pretty opaque, nearly opaque. So I instead turned to the scanning electron microscope. So I take some of this, macerated gunk from the hydrofluoric acid maceration, stick it on a slide. And it's kind of laborious, but I can sort of find fossils. A lot of this is gunk. But the fossils I can sort of identify and take pictures of and then leave little Sharpie marks and then move to the SEM. And if you can see here, again, like a lot of this is just crap. I think it's my technical term. But there are a lot of these little circular disks. Okay. And if you look at them up close, you can see all sorts of amazing detail. And so I started in on this project thinking I wanted to really understand the morphology of these forms and maybe try to figure out what type of eukaryote they are. We don't know. Again, we know they're eukaryotes, but what kind, we don't know. And found some interesting, evidence that some of the species were oversplit or lumped, and so worked on the taxonomy. But while I was doing that, I was looking up close. And I started to notice that there were I'd start seeing these perfect holes in some of them. And they were in different species. So here, an interesting morphology of the fossil, but then they have these perfectly circular holes. And they weren't on every fossil, but they were common enough. And I did a section through this fossil, through the hole. And you can see the hole doesn't pierce the inner ornament. So it's not coming from the inside out. It's coming from the outside. It's beveled. It's just through this outer envelope. Here's another example, another specimen. Here's a fragment of a fossil, but you can make out 3 of these holes, again, kind of beveled. And so then I started recording, okay, in each sample, in an individual fossil specimen, what are the sizes of the holes that I see? And so the y-axis here is the hole diameter, and I saw them in 9 different samples. And the different colors here are different species. And a couple sort of takeaways from this graph is that, first of all, they're found in a variety of different species, and species that I have no reason to think are closely related to each other. There's sometimes one, but often multiple holes in any individual specimen. And there seems to be some kind of specificity where certain species, like these red species and the one I've indicated in orange, have larger holes. And the other species just have this very sort of 0.1 to 0.6 micron, very small hole diameter. Interestingly, that corresponds to the size of of the species, that these are large species that are getting the large holes. So, the one obvious question to ask is, well, is this just a biological character of these species? Are they pores? And we know that there are fossils and modern organisms that have pores in the walls. But I thought the fact that they're found in so many different species that are clearly unrelated, and they're not found in every specimen, or even a majority of specimens of any given species, suggests that it's not a biological character. And they're randomly distributed as well on the fossils where they occur. Another possibility is, could they be formed by mineral punctures? Well, I have an example of what mineral punctures look like, right? And they're not the perfectly circular holes in an otherwise, beautifully preserved fossil wall. Another thing I struggled with is, well, maybe it's microbial degradation of the wall. In other words, these are fossils. The organisms, they die, they sink to the bottom, or maybe they were living in the sediments, and then bacteria are going to eat them up, of course. So I got a little bit into the literature of, well, what does microbial degradation look like? And it turns out there's this whole literature of people sticking plant cells into rumen, cow rumen, to see how they degrade and see what the bacteria do. And what you can see is that it's they sort of erode the whole surface and they make these irregular pockets. And I was also thinking, if it's microbial degradation, I wouldn't see just these individual holes. I would see specimens that had evidence had further down the path of degradation, larger holes, patches. I only saw these perfect little holes. And I knew from my work on the testate amoebae that there were amoebae out there that actually drilled into their prey. And they're known as vampire amoebae, vampirellid amoebae. And I'm going to go over time, but I'm going to show you this video here of what this looks like. And there's this creepy music to go along with it. Right? So they're common in soils. They're actually also known in marine environments. And what they do is they will sort of partially engulf their prey and put their sort of pseudopod right up against the prey wall and presumably release some kind of enzyme that eats away. And here you can see this amoeba has just punctured and it's sucking up the insides. And now it's punctured the other wall here and it's making its way through. I mean, there's terrible stories here of how they stick their pseudopods in and pinch off. And I'm just going to show you this one too. It's pretty cool. So this is sped up like 500 times. But they're a real problem in algal modern ponds where they're trying to grow biofuel, algae for biofuel. These parasites will come and just destroy the whole crop of algae. So you can see the algae here, right? And then you can see they're sort of ghost-like moving around. And those are the vampire amoebae. And they engulf, you can see that there. Make multiple punctures, stick its pseudopods in, and actually form a cyst itself, multiply inside, and then you just saw all the babies go off. Again, the scary music, right? It's terrifying. It's terrifying too. All right. So here's an example of an amoeba with attacking these fungal spores. They were first discovered in soils. This is an up close that's the amoeba cytoplasm. It's eroding a circle in the fungal wall, and then it's almost like a manhole cover. It lifts the circle out. It actually swallows it, and then it sticks its pseudopods in and starts pinching off the cytoplasm inside. And here's some of the holes, just for comparison with my fossils. And they're species specific, and these vampires even apparently have different species prefer different food types. They're picky about who they want to eat. So Theratromyxa makes these tiny holes, 0.2 to 0.3 microns in size on these fungal spores. Vampirella makes holes of much more variable in size. You can see here where it's like that manhole cover, right? It's in size, and it's going to lift that up. Now, at the time that I was sort of putting this together with the vampire amoebae, I thought back to my paper on the vase-shaped microfossils from when I was a PhD student. And at the time, I had reported these bizarre semicircular holes in, again, different species of VSMs. They're not present in every single one, every specimen of a particular species. I don't think they're biological. But you can see they're all about the same size. Here's an example of this semicircular hole, semicircular here. Here's another one. What's interesting is the long axis always seems to be parallel to the long axis of the test. And I have been showing these pictures for years, hoping someone will tell me, oh, I know what made this. And I still don't know. But all I can guess is it's maybe one of these predators, which drilled into the wall. So I went back and looked at these pictures. And I realized, I don't know how I totally missed this, but some of these specimens have circular holes, perfectly circular holes. You can see it really well here. Here. These are much larger, right? There's a scale bar, than the tiny holes in the organic walled microfossils. But I think it's perhaps the same kind of thing going on. It's a drilling predator. Okay, so I thought, okay, awesome. We have evidence of predation, and we have evidence of this vampire amoebae. And we know the vampire amoebae, we know where they are in the tree. They're in this group with the foraminifera and the radiolaria. So we can say, this clade was around at this time. The problem is the more I got into the literature, I realized that this type of drilling or piercing and sucking out the insides is all over the tree. So there are some slime molds that do it, for example. There are some of our close relatives do this. And there are foraminifera that do it, and some other groups also in the rhizaria. So, it's either something maybe that was present in the last common ancestor or something that evolved over and over again, this prey type strategy. So I can't say, where in the eukaryotic tree it is. And in fact, it turns out there are these bacteria that we know of. They have this wolf pack behavior where they'll move around in a swarm and attack a victim and leave these different holes. So it's possible, possible these holes maybe were made by bacteria. I think it's less likely. But one thing we can say, even though we don't know who the vampires were, we do know who the victims were. And we know that they were eukaryotes. So we can say, all right, in the Chuar group, so far, maybe there's going to be new evidence that comes to light. It looks like we have the oldest evidence for predation on eukaryotes. So just to summarize it, it's those drill holes, and not only predation, but a variety of different predators, right? So we have whoever's making those semicircular holes, whoever's making those large circular holes in the VSMs, whoever is making those tiny 0.1 micron holes in the organic walled microfossils, and probably a different species making the 1 micron size. And the vase-shaped microfossils themselves are evidence of predation at this time. And also, in the last 2 years, a paper was published, biomarker evidence from the Walcott, the uppermost part of the Chuar, where they reported a new biomarker that they said looks very similar to molecules made today in sponges that protect the sponge itself from toxins that the sponge makes that will rupture eukaryotic cells. And the idea is the sponge itself is poisonous, and so someone is eaten, so the sponge makes these toxins to protect itself from getting eaten. And so since it's making these dangerous toxins, it needs some way to protect itself. So it makes these compounds. And so, this paper said possible sponges in the Chuar group, but I think they backed off a little bit and said, at the very least, we have evidence for probably predation on eukaryotes. So a eukaryote making these structures to protect itself from the toxic, poisonous compounds it's making, if you followed that twist. Okay, so of course, you may be asking yourself, awesome, there's evidence for predation, so what? Who cares? And the reason it might be important is back to this larger question that I'm interested in, which is the rise of this complex life, right? We were billions of years on this planet. We're certainly life diversified, but it remained simple. I mean, there wouldn't be anything interesting to see if you visited the planet. So the sort of widely held view at the moment for why eukaryotes diversified from 1.6 to 800 million years ago is that it has to do with oxygen. But we know, sort of generally speaking, from various oxygen proxies that probably oxygen rose sometime during the Neoproterozoic. And if I just grabbed a couple of examples of this type of thinking. The idea is that, well, eukaryotes are aerobic, so they're restricted to oxygenated environments. And if oceans were predominantly anoxic at this time, and perhaps harbored large pockets of hydrogen sulfide, which drew down bioessential elements, overall, along with the low oxygen availability, this challenged the emergence and diversification of eukaryotic organisms. So the idea is once oxic environments expanded, eukaryotes expanded. Here's just another quote, similar. If periodic anoxia was common throughout shallow oceans during the Neoproterozoic, all eukaryotic life would have been oxygen stressed on evolutionary timescales. So this may explain why we had eukaryotes at 1.6, but we don't get an expansion until like 800 million years ago. Okay, so great. This all sort of makes sense. The problem is that if you actually dig into the literature on modern protists, modern microbial eukaryotes, you find that eukaryotes are not oxygen stressed. I mean, it depends on the species. That we're eukaryotes, we need oxygen, but different eukaryotes need different amounts of oxygen, and some actually are restricted to anoxic environments and can't live in oxygenated environments. So realize this is confusing. I just put this up here to show you, to hone in on the colors. These are samples from various different anoxic basins around the world, and they're sampling of sequences. Right? So can't see the organisms, but just the sequences. And what this reports is this incredible diversity of eukaryotes. All the different major groups of eukaryotes, including algae, are present in these anoxic environments. And there are additional studies that show that protists and fungi can be actually quite abundant in these environments. This is just an example from the water column in the Cariaco Basin showing that flagellate cells peak below the chemocline, probably feeding off of all of these bacteria that are making a living off of the chemocline. There's other examples of really abundant protists in sediments, in anoxic sediments. And it's looking like a common thing among these protists is that they are associated with bacteria that either live outside their cells or inside their cells, these bacteria or archaea, and that these prokaryotes essentially enable these protists to live in these different environments. So it's sort of like us being able to live in Antarctica. If we have the right environment, we can alter our environment just right and live there. These protists are doing the same thing. They can detoxify the waters. They can get oxygen from cyanobacteria that live nearby, and they can get food and so on. Okay, so I said to myself, well, so this idea that rising oxygen levels meant eukaryotes could expand is sort of against this finding that eukaryotes actually do just fine, some of them, in anoxic settings. So it reminded me of this paper by Eric Sperling and others that came out a couple years ago that argued that oxygen has been invoked for the Cambrian explosion of animals. And he said, the problem with this idea is that if you look at anoxic settings today, they are low oxygen, hypoxic settings. There's actually a lot of animals there. But what we don't see are carnivores. So if we look in the environments where there's no oxygen at all, there's no carnivores. Plenty of animals, but those animals are grazing on bacteria. They're not eating each other. And I thought, so what Sperling says is, maybe the Cambrian explosion, we have this sort of false dichotomy where it has also been suggested that predators carnivores caused the Cambrian explosion. And they argued, maybe it's oxygen and carnivores, that rising oxygen allowed the appearance of carnivores, which then drove the Cambrian explosion. And it makes sense that predators would have a big influence on driving diversity. We know that from modern experiments. that they can increase the diversity of their prey. It makes sense that predators maybe were driving the Cambrian explosion. If we look at the first appearances of skeletons in animals, they're concentrated here in the early Cambrian. If we look at some of the earliest skeletons, they appear to be adapted for predation as protection against predation. And of course, large size has long been argued. The evolution of large size has been argued as a defense, maybe against predation. So maybe that's why the sauropods got so big. So if we take this view to predators then in oxygen, it's interesting to note if we look at these anoxic environments, they're teeming with protists, but there's few predators as far as we know for some reason. And perhaps rising oxygen then in the Neoproterozoic allowed tiny predators, vampires and so on, to appear, and that they then drove this diversification, this evolution of multicellularity. I'll just skip through that. And these morphological innovations, like this armor, these scales that probably armored a protestin cell. So, there's this potentially interesting sort of parallelism in the history of life here, where the rise of eukaryotes maybe was driven in part by the rise of eukaryotic predation on eukaryotes, and the Cambrian explosion was driven by a rise of carnivores. I don't know if I have time to get into that. You can ask me about it later if you want. So just sort of the takeaway here is that we can see in the Chuar Group 730 million years ago that it wasn't boring. Life was pretty complex. There was an incredible diversity among protists. There's evidence for a number of predators, some of those predators preying on other predators, so pretty complex food webs. And it's possible, this is speculation here, it's possible that maybe predation is driving this expansion of eukaryotes that we see in the Neoproterozoic, and that maybe predation itself is tied to the evolution of the atmosphere and the rise of oxygen. So repeated patterns in the history of life. Thank you.
Thank you very much.
Yeah, sorry, I took a little liberty with the time.
It was a fascinating story. First, I'm going to give you this amazing swag bag that I crushed you earlier. So thank you very much for coming. And I'll open it up for questions.
Awesome.
Oh, sorry. Go ahead. Yeah.
I just wanted to make sure everyone knew that we had a reception that's going to follow the talk down in the Silver Family Geology Museum. That's down the hall around to the left, and there's lots of goodies there. So make sure you stick around for that.
Great. All right. Thank you, Peter. So do we have any questions for Susannah? And we can continue them. You can.
Oh, okay. Yeah. I forgot your name. Sorry, Jeff? Andy? Yeah.
So, as I'm sure you know, there are also many different types of microbialites in the Chuar.
Yes.
So I saw, 5 to 10 different types. So do you see, what is the relationship between these with the microbes in the Chuar or in general, I guess?
So I didn't show you pictures because they're not as well preserved, but those vase-shaped microfossils, I've also found them in silicified microbially laminated carbonates, and they're really abundant. And that's actually not as abundant as those nodules, but you can see them lying along the laminae, and this is actually similar to where they've been found elsewhere around the world. They seem to really like organic-rich settings. We know this from modern environments too. They like the really organic-rich settings. So they were probably, I suspect they were living there, grazing on the mats, maybe. Yeah. Yeah, Maya.
You suggested that they might have been benthic and not floating in the water column. You think that they're on the seafloor?
So that it's based on analogy with the modern, which, again, like what we know about protists, in 10 years from now, it's going to be so much more, right? So, but what we know about these testate amoebae today, we find them in thin films of water on the surfaces of mosses, or in soils, in among the leaf litter. One of the most common, like more common than earthworms, they're much tinier. Very common in soils. And so they sort of, I mean, they're walking along surfaces, in other words. They're not floating. But you know what the analogy is in the Chuar group, maybe they're on these surfaces of stromatolites. The nodules, where there are trillions of them, I don't think that was, you know, they weren't living there. I suspect that they were transported. And we know, again, from the modern that those tests are really robust. They can act as like little sand grains bouncing down the rivers and getting deposited in beaches. So probably transported to where I found them. Yeah, behind Maya.
Your diversity graphs showed fairly high diversities of these eukaryotes before Snowball Earth.
Yeah.
And then immediately after Snowball Earth, which is a blank spot I indicated, the diversities are even higher, much higher.
Yes.
So could you, what's the similarities and differences between those assemblages and the ones that preceded Snowball Earth?
They're totally different.
Ah, okay.
Not the same species at all.
A lot of evolution has been going on during glaciations.
Yeah, or, you know, I mean, the problem is the record isn't good enough. You might be able to say, oh, there's a mass extinction with the glaciations. And I had a PhD student who looked at the shales in between the two big Snowball Earth glaciations, and she found fossils, but nothing, no diversity. Very simple, smooth-walled, kind of boring forms. So that suggests they're gone some point, but you know what's interesting is if you go back, and we were trying to make this case. I don't know if we've convinced anybody, but if we go back to that more recent diversity curve that I showed you, where I showed that VSM peak, you can see that those species are starting to decline, and that they're pretty much mostly gone. And then the VSMs take over, and then they're gone. So, you know, maybe it's a gradual extinction, maybe it's a senior ellipse effect type thing, and they disappear around the same time. I don't know. But yeah, this is a totally different biota.
Are there important new groups of eukaryotes that appear for the first time after the glaciations? Or just kind of more?
We'll get most of these, a lot of these microfossils, we just don't, we know they're eukaryotes, but we don't know where they are. But it is true that in the Ediacaran, like some of the beautifully preserved seaweed biotas, there's evidence for brown algae, the earliest brown algae, the earliest really convincing evidence of animals, maybe the biomarkers a little bit earlier. But yeah, in terms of just those organic world microfossil species, we don't know what kind of eukaryotes they are, but it's a totally different group. And no continuity. Yeah.
Can you identify young and old species in terms of age? What I'm driving at is if the holes are done after death, there should be about the same density of holes, regardless of age stage. But
right.
If done because of predation while alive, then older ones should sustain more hosts than younger ones.
There's no, there's no growth like that. You know, not like, right, with, say, you could say a baby clam versus a larger clam growth lines. It doesn't work that way. They make I mean, probably a lot of what you were seeing are cysts, are the resistant walls that they made when they were sort of hibernating, and those are made all at once. So no, yeah, and then how long were they out there floating? And I don't think that's a testable idea.
Great. Well, thank you very much again. Let's go.